Students, welcome to the Gyan Megh e-content platform of Institute for Excellence in Higher Education. In the Faculty of Science, in this e-content, we will be discussing for BSc semester 2nd under the subject Biotechnology, Cell and Molecular Biology. The course is Major MJS 285. In this e-content, we will be covering the prerequisite for this topic, previous learning, objectives, introduction, key experiments to prove DNA as the genetic material, Griffith transformation experiment of 1928, Avery MacLeod and McCarthy's experiment 1944, Hershey and Chase experiment 1952, and then we will summarize and some of the extended learning resources are given. After studying this lesson, students, you will be able to illustrate DNA as the genetic material, describe the Griffith's transformation experiment, describe the Avery, MacLeod and McCarthy's experiment, as well as describe the Hershey-Chase experiment. For studying this module, the prerequisites are knowledge of heredity, knowledge of general structure of cell and its composition, and general knowledge of life history of bacteria and viruses. Possibly these things you will be knowing after studying in the fourth semester. You have already studied basics and history of heredity, role of DNA and RNA, and constitution of cell. The discovery that DNA is the genetic material was a landmark in the history of biology. This realization paved the way for understanding the molecular basis of inheritance and laid the foundation for the fields of molecular biology and genetics. The journey to unravel the mystery of genetic material involved in several key experiments and several work of the scientists. 3 experiments and discoveries collectively established the proof of DNA as the genetic material that led the identification of DNA as the genetic material. Griffith's transformation experiment in 1928 was first among them. Frederick Griffith conducted an experiment with bacteria that provided the first hint that DNA might be involved in the genetic material. He worked with 2 strains of the bacterium Streptococcus pneumoniae, a virulent disease-causing strain and a non-virulent strains. Griffith observed that when he injected mice with the heat-killed virulent strains, along with the live non-virulent strain, the mice died and the live virulent bacteria were recovered from their bodies. This phenomenon termed as transformation, which suggested that the something from the heat-killed bacteria was transferred to the live bacteria, transforming them into virulent form. However, the nature of this transforming substance was not clear at that time. You can see here in the graphics that when the virulent S strain was injected to the live mice died. When the non-virulent R strain was injected to the mice, then he found that the mice was alive. When he heat killed the S strain, that is the virulent S strain heat killed, which retained the shape of the bacteria, and then he injected mice with this strain and he found that mice was alive. Then he performed an experiment in which the mixture of heat-killed strain and live R strain was used. When this mixture was injected to mice, the mice died. And when he recovered the bacteria from the died mice, when he applied the live R strain and heat-killed S strain, he found that These contains the virulent S strains, which proved the fact that the bacteria which was heat killed transferred something from their body to the live R strain and transferred them to the live R strain, and this process was known as transformation, and it was observed that this is the genetic material which carries information for these things. Building on Griffith's work, Oswald Avery, Colin MacLeod, and Maclean McCarthy aimed to identify the transforming substance. They used an enzyme to selectively destroy different components of the heat-killed virulent bacteria such as proteins, RNA, and DNA. Only when DNA was destroyed did the transforming ability disappear. This provided a strong evidence that DNA was the substance responsible for genetic transformation. The Avery, MacLeod and McCarthy experiment conducted in 1944 played a fundamental role in identifying DNA as the substance responsible for genetic transformation. As stated, building on Frederick Griffith's work, Oswald Avery, Colin MacLeod and Maclean McCarthy aimed to pinpoint the specific component in the heat-killed virulent bacteria that caused the transformation of the non-virulent strains. The primary objective was to determine which component of the heat-killed S strain, that is the virulent strain of the bacteria, was responsible for the transformation of the R strain, that is the non-virulent strain. The researchers used the bacterium Streptococcus pneumoniae and continued with S and R strains as in the Griffith experiment. They treated the heat-killed S strain with enzymes that specifically targeted different biological molecules like proteins, RNA, and DNA. Enzymes that destroyed proteins were used and the treated mixture was tested for its ability to transform the R strain. Enzymes that destroyed RNA were used and the treated mixture was tested similarly. Enzymes that destroyed DNA were used and once again the mixture was treated for the test of transformation. Here you can say that a smooth-walled bacteria in liquid culture medium was taken and then it was transferred to the solution in the test tube. Then they centrifuged it, and after centrifuging the S cell spun were recovered. Now they were heat killed and cells extracted and opened, and after that cells filtrate slaved in test tube and cells were discarded. Now transforming power of all the material which was just transformed was tested, and here they transform in the process of transforming power testing. When they used protease, they still having transforming power. When they used RNAs, they still having transforming power. But when DNAs was used, then there was no transforming power, which suggests that when protein was disabled, they have their transforming power. When RNAs was disabled, they have their transforming power, meaning by that protein and RNA is not the genetic material, but when DNA was used, the transforming power was lost. So when treated with protease or RNase, the transforming ability persisted. This indicated that neither proteins nor RNA were the transforming substances. However, when the mixture was treated with DNAs, the transforming ability was lost. This suggested that the destruction of DNA prevented the R strain from being transformed into the virulent form. Due to this observation, Avery, MacLeod and McCarthy concluded that DNA was the substance responsible for the transformation observed in the Griffith experiment. The transformation did not occur when DNA was enzymatically destroyed, providing strong evidence that DNA and not proteins or RNA was the genetic material. In the third experiment, Martha Chase and Alfred Harsey studied bacteriophages, viruses that infect bacteria. They used the T2 bacteriophage, which consists of a protein coat and DNA core. In their experiment, they radioactively labeled the viral DNA with phosphorus 32P and the protein coat with sulfur 35S. After allowing the bacteriophages to infect bacteria, they found that only the labeled DNA, not the protein, entered the bacterial cells and directed the production of new phages. Here you can see on your screen that when T2 phase, you have already studied the structure of T2 phases. These T2 phases in first culture A was labeled with radioactive 35S and normal 31P, that is protein coat was labeled. The bacteria in medium of the labeled protein with the labeled DNA. When bacteriophages were infected to the bacterium, the DNA entered inside the bacterium, they multiply and the bacteriophages were recovered. These bacteriophages, progeny phases were unlabeled. Now, in the culture B, they have used labeled DNA with 32P and normal 32S bacteria in the medium of unlabeled protein with labeled DNA. When these bacteriophages were infected to the bacteria and the DNA entered inside the bacteria, multiplied, and the recovered bacteriophages were labeled with the DNA, this shows that the DNA is the genetic material, not the protein. If we summarize all the three experiments, Frederick Griffith's experiment was a groundbreaking step in understanding the nature of the genetic material. He worked with two strains of the bacterium Streptococcus pneumoniae, the virulent disease-causing S strain and the non-virulent R strain. Griffith's experiment was groundbreaking because it provided the first evidence of a transformable hereditary material that could change the phenotype of an organism. Building on Griffith's finding, Avery, MacLeod and McCarthy aimed to identify the transforming substance. They used an enzyme to selectively destroy different components of the heat-killed S strain, including proteins, RNA and DNA. When they destroyed the proteins and RNA, the transforming power was retained, but when they treated with the mixture with an enzyme that destroyed DNA, the transformation did not occur. This strongly suggests that DNA was the substance responsible for the transformation of the R strains into the virulent S strain. The Harsey and Chase experiment provided additional evidence supporting DNA as the genetic material, and it focused on bacteriophages, viruses that infect bacteria. They found that only the radioactive DNA entered the bacterial cells, not the protein. This indicated that it was the DNA, not the protein, that carried the genetic information. So DNA is the genetic material. For further reading, you can refer these textbooks: The Cell, A Molecular Approach, 5th edition by Cooper and Hussman; Molecular Biology of the Gene, 6th edition by J.D. Watson, Baker, Bell, Gann, Levine and Losick; and then Genetics by Peter and Principle of Genetics, Sunstead and Simmons. You can also refer the reference book that is Molecular Biology, Concepts and Experiments, Cell and Molecular Biology, 8th edition, and The World of the Cell by Pearson, Benjamin Cummins. You can also refer the video resources. These are the links which is given on your screen, as well as you can refer the text resources. Now assess yourself. How did Avery, MacLeod and McCarthy contribute to the proof of DNA as the genetic material? The options are: they discovered the structure of DNA, they demonstrated that DNA could transform bacteria, they isolated RNA from cells, they observed DNA replication. The answer is they demonstrated that DNA could transform bacteria. Second question, which experiment involved injecting mice with the different components of the bacterial cells to determine the transforming principle? The options are Hershey and Chase experiment, Griffith's experiment, Michelson-Stahl experiment or Chertoff's experiment. The answer is Griffith's experiment. In the Hershey and Chase experiment, what was leveled with radioactive material to trace the genetic material? The option is proteins, DNA, RNA, lipids. The answer is protein. So students, you might have understand all the three experiments that are the proof of DNA as the genetic material. In next e-content, we will discuss another new chapter of BSc second semester. Thank you.